Welcome all to this session, which is a report back from all the different breakout sessions. And we have approximately 10 minutes for each of the breakouts, so 5 to 7 minute talk with some time for question. And the first breakout session we have is for the gut, and Paul Klennerman from Oxford University is going to start.
Thanks. Right, so we have a slide. I hope it will help. Not that one. I think these. Just wait a sec. There's something that wasn't on the slide while we're waiting for it to appear, which was about cell, or rather biopsy storage using a freezing protocol, and then disaggregation when you need to use the cells for a downstream application, which generally seemed to be something people were taking up because it gives you a lot more control over the experiments. And the comment was made about duration of storage, and after a period of time, there may be some degradation, although it seems to be really pretty robust, actually, and you might move on to using nuclear sequencing in later samples, but that would allow people to get a bit of control over their experiments. Right. Here we are. Good. These were the things that sort of took up our time. So the gut, as we heard this earlier in the day, is big, and it's complicated and full of microbiome, just to add to the diversity. And so we need to define the normality as best as possible. In other words, there was a lot of discussion about health and disease, but we want to define health really well for its own sake, because there's going to teach us a lot about homeostasis and cell networks, and it will also give us a good basis to understand disease, and I think also vice versa, the disease might give us some insight into normal function. So normality, there isn't a perfect way of doing this, because each individual technique, if you're going to use organ donors, they're not completely healthy. Biopsies don't get you all the way through to areas which might be important, and not everybody gets a biopsy. So there wasn't any kind of perfect method, but we figured that probably combining approaches is going to be the best way of getting a glimpse of reality. There was a lot of discussion, and this might be relatively specific to the gut, but there may be resonance in other sessions about defining location as best as possible. So up to a point, this is the responsibility of the people taking the samples, and everybody in the food chain, basically, from the endoscopist or the surgeon or the pathologist down to the people loading it up onto the portal, need to keep a track, because there is variability, and otherwise we're not going to be able to compare things. It's difficult enough anyway, but if you don't even compare the right part of the gut. We were hopeful, and this is an experiment, I think, to be tested, that you could actually use the information that was coming from these very, very well-prepared initial samples. For example, Sarah Teichmann's group are going to be looking up and down the large and small bowel in the organ donors. You might be able to find a sort of molecular gradient that gives you an address for either the whole biopsy or cells within the biopsy that could then, in subsequent iterations, allow you to sort of somewhat either cross-corroborate or even bypass the need to give a very precise biopsy site, because it would all be embedded in the sequence, basically. But that needs to be tested. That would be fantastic and interesting for its own sake. Rare cells, there was a bit of discussion about this, but we agreed that rare cells can be really important. We can define what we mean by rare, but there'll always be cell types which are relatively infrequent, which we would like to sample. So we do need to get to some level of depth, otherwise we're going to be potentially missing the cells of most interest, again, either in health or disease. I think we would like, as a group, to study disease in parallel with normality. That's partly because of people's drivers to get tissue, people's interest in disease. But the issue with the diseases is that they tend to be a little bit opportunistic depending on the site. So we're only going to be looking at essentially fractions of the gut. And again, it lent itself back really to this sort of loop back to this point that we do need a sort of solid atlas in normality, which is well anatomically annotated. So all the more reason for doing that. And if we can get a range of normals, which would incorporate genetic variants, age, and so forth, and ideally also microbiomes ultimately, that would be great. But just to get off the ground, just to simply get the normals in the numbers that people can achieve with the funding that's available. So this point was made that in pathology you might get frequency changes or transcriptomic changes within a population or both, and we need to just think about both when you're interpreting change. Is it frequency or transcriptomics? If I think made the point, you might want to elaborate on that. Bulk analyses, we'll agree to have a big role to play. They're relatively routinely done and will anchor all of this, especially these issues about normality and anatomy. We did feel as a group, and there was really no dissent, I think it's obvious, that spatial approaches will have a big role to play in dissecting out the function of these cells. We like to think that the GI tract in general, specifically the luminal, is going to be a very good place to do this because there's an abundance of tissue. People get either large chunks taken out or their biopsies are repetitively done, also longitudinally. So it would give you an opportunity to sort of explore this and define what's best practice, and we would like to put our hands up to be the leaders for the spatial approaches. And I think this is really probably a very important point, which we didn't get to the end of, but ultimately we would like to define function both in health and obviously dysfunction in disease. That isn't going to come just from this initially, but does need to be addressed, I think, if we want to get the best out of this version of the Atlas. Even at this stage, we should be able to make some new, derive some new insights into function. But that will need incorporation of the spatial data to understand who's talking to whom, where, and ideally some host genetics to understand the role of that variability in ranges of normal or in disease. So I think that's really everything. So I'll pass over to questions, and Gabrielle is going to help me answer them as well.
Great. Thank you very much, Paul. Are there any questions or comments for Paul?
No. Good. Yeah.
Oh, Abby. Oh, sorry.
Sorry, because of the streaming, people change Since I was in the session, I actually know that was a tough one. So the last one with the function that goes back to Dana's point from her talk, could you say a little bit more about where your thoughts are and how we could get there? What would function look like?
Okay, well, I think there are different components to that. So I guess our approach in our lab has been around immunological function. And then you need to see groups of cells talking to each other. Classically, to understand the function in that setting, you would need a knockout, and then you could really assess what the non-redundant function of that cell was, or that cell, that gene was, I suppose. So we may be able to do that with, and this would need a lot of discussion, with the natural genetic variability in human populations. If you could scale it up, map those as you're already doing onto individual cell types, and then see how that disrupts networks of cells, which are ideally in this scenario spatially related. So if you could pull all of those things together, you would get an idea of function in human tissue, rather than having to kind of do some reductionist experiment in a mouse model, which would be perfect in my view. So I think that's what I had in mind. Now, for the physiological functions of the barrier in terms of, tight junctions, solute transport and things like that, I'm not sure I can really answer that question at this point. Those are going to be equally important.
Right, we're going to have one more question from Katrina.
Thanks, Paul and Garabet. Thinking back to Orit's talk earlier in the day when she presented this lovely roadmap, I was wondering, did you talk about how other groups might align to that, or is everyone aligned, or is it kind of too early because people are trying out different approaches still and different methods?
We didn't explicitly try and align it with what you described, although I think it was sort of inherent in everybody's approach. Not everybody's being as comprehensive as that. Probably the Sanger group are ultimately, but I think that all those ideas are sort of very similar to what we discussed, with the additional dimension of disease, which I think everybody felt we had an important role to play, both as a motivator and also as a comparator. I don't know if you want to add to that, Gavin.
No, I think we just touched upon sort of the depth and the breadth of sampling. So that is a component that I think aligns nicely with what Ori presented earlier, and hopefully we're going to get the community align, or at least join forces to move forward with this approach. So that's something that we touched upon, but we haven't discussed it more. And something that I can eventually disclose, but not fully, is that we do have efforts from Hemsley to build the gut cell atlas, starting with the terminal ileum and ascending colon, which we will be announcing very, very soon. However, one thing that is important to note is that this is a cohesive effort. We're thinking about multiple aspects of building sort of a roadmap and building and implementing an atlas. So we're hoping that this effort will be replicated in other tissues and organs. So stay tuned.
Great. And on that note, I think we're best to move on. And so we next have Chloe Villani, who's going to give us the summary from the immune breakout session.
Sounds good, yes. So I picked the short straw. I'm talking in the name of Holger and Effie. And I do want to say to the organizers, for next HCA meeting, it would be really good to do the report the next day, because I'm afraid that we're not going to do justice at all the amazing feedback we had in session. This was our layout. We're very organized. And so we wanted to follow up on Dana's question about the question we want to answer. Don't be too disappointed, Dana. But also like what the roadmap looks like, what would be our first draft, right? 1.0 of the ICA. Does disease matters? How do we tackle this? What are the challenges in terms of data harmonization, which is a challenge that is very particular to the immune cell atlas effort, because of course immune cells are found across all organs. And is there anything we can do to better empower the ICA community moving forward? Okay, so in terms of the roadmap of who's doing what, we invited the audience to tell us a little bit about the different efforts that are going on, thinking that this can help moving forward in better synergizing. And it was interesting because we had both lineage-specific effort, which is very important in the context of doing a deep dive to really define the full extent of cell states across tissue. But there was also a tissue-specific effort, and more this particular HCA meeting, we heard actually from at least 3 different groups that are collecting multiple tissues from a single individual. So there's probably even more effort, but at least we heard from 3, which is actually very important when we think about trying to harmonize cell type and cell state identity across tissues. There's, of course, a computational component, but there's also an experimental design component that can help in mitigating some of these technical artifacts. We heard also a lot about groups that are taking into consideration ethnic background, which is also a very important consideration. Lots of blood effort, and there's actually a few efforts that are not on healthy tissues, but are immediately on disease efforts. So we heard about pediatric cancers, different type of immunotherapy-related efforts. I'm going to do the shot for the one person that works on mesenchymal cells in the bone marrow. He was wondering if there's anybody else that does it. So if so, please Slack, and then you can meet. And there were also efforts about organoids, recognizing that it's not organoids with immune cells, but that would be an important setup moving forward for functional studies, as well as in vitro reprogramming models towards generating immune cells. So that's why they're interested by the immune cell atlas effort, to be able to benchmark their effort, and that's in the context of organ transplantation. There were a few comments about challenges which are kind of pretty obvious, but we thought we should at least summarize them. So problems of reproducibility, and some of it has to between our different ICE effort, and some of it is actually because of the way we sample the tissue. And so it might just be a sample size issue at this point. Immune plasticity is, and so that's a problem specific to the immune cell atlas effort. We have a range of cell states, and some of it can be triggered by different perturbations. So how do we do with better capturing this full plasticity? We didn't resolve that question, but it was brought up. Demographic variations. We've tackled tissue sampling questions in previous meetings, but it came up again. We were missing some important cell types because of the way immune cell types because of the way we're processing tissues, and that we should keep an eye for this. There were at least 4 people that brought up the fact that we don't really know what's normal, and we know what's looks like normal, and we know what looks like disease, but there's actually some individuals we're collecting from. They may have some ongoing viral infections or be carrying CNV, so we're not taking into account the virome, and that can lead to different type of outliers, and that we should consider really analyzing the full spectrum of individuals, not just the healthy, and the very diseased, but actually the in-between. In terms of, sorry, we summarized the thought. This may look a little small in terms of font, but like what the first draft looks like. Dana, this is very wordy, but I promise you the next slide summarizes it better. And so we couldn't really agree on the number of individuals we needed. There actually was some interesting arguments in the room about, do we actually need disease for the first draft? Some people say, absolutely, you don't even need, some people say, we don't even need healthy. You just need to compare different diseases to figure out what's specific to a disease. And some others said, no, all the effort should go to really deep reference health atlas. Some people said the magic number is 1 million. Holger, I'm going to look at you, because we were forcing at like what's the good number to kind of establish a reference set. But back to Avi's point this morning, defining it statistically as of the frequency of cells we want to capture, like 5% is probably the way to go. So you see this repetitively. Perturbation. A lot of people said perturbation is essential to understand the function of tissue, so we should definitely include this. We need to better coordinate as part of this HCA effort with the other organ-specific atlas, because all of them are capturing immune cells, and perhaps we should generate better guidelines in terms of what the ICA would need so we can synergize. We believe that 2.0 version should perform a deeper dive in the rare cell types, and that version 1.0 can help nominate the key surface markers that could lead to developing a more uniform site-seq panel that all ICA members could use moving forward. And that we should focus on the easier samples to access and leave the harder samples like pediatric samples for next generation. So what does it look like for a first draft? I should make sure we capture the wide range of ethnic backgrounds, so there's a great Asian Pacific effort in that regard, and all samples should be genotyped, should also capture geographical environment. We should make sure that we report the range of frequency of cell types, which will be essential for disease analysis. Some people should say that should be our main milestones. We argue around how many atlases should we have? Should it be one or should it be multiple? Should it be PBMCs, tissues, pediatric adults? Should we try to combine it all? We don't have an answer. I think we just need to try it. 5% seems to be a reasonable number as a threshold for cell states. And making sure we integrate all the organ-specific efforts, immune cells in our data sets, and that integration will allow us defining what cell types and states we should be focusing on in the next effort. We debated on the disease to prioritize. There's no specific answer, and that goes back to the need to defining a more normal, healthy states. But that inflammatory states should be part of 1.0 version because they are part of what defines healthy. In terms of integration, the same thing came up as previous sessions, previous meetings. We need metadata, all sort of metadata to define what's healthy and help integration. And there was an interesting discussion between different computational folks in the meeting, so I invite those to come in back, because maybe we didn't summarize it well. But they said that we need to use replicates to define the null distributions and need to define the range of confidence. And they didn't really define what they meant by confidence to establish what is a true stable cell states. And some have argued that we should analyze all data sets separately, annotate them before merging them in the fear of losing biology through integration. Dana, I'm just reporting, so don't get mad about the next one, okay? Some people have argued that in the context of the healthy atlas, immune cells, the batch effect may not be so bad for integration as compared to disease. And so integration across organs may be pretty straightforward in terms of the batch correction. But in the context of disease, it will be really hard to know if it's biological variation or or technical variation. And so the proposal was to try to focus on integration on the healthy states to start with. And that we just don't have enough cells right now to know if we have real problem or not. And that we should think beyond just a molecular definition for cell states, but try to think of different features to define the cells. We did talk a little bit about multi-omics integration, and obviously people just want everything if they were unlimited funds. So moving forward, at least 60% of the room said by show of hands that an ICA virtual meeting would be really great to share unpublished data, try to better synergize moving forward, that it would be really helpful to have an IC portal that would help the greater immunologist community to connect with us and integrate some of the classical flow data so they could relate better with our data, and that we probably need a more specific working group for data integration and interpretation. And a representative from the DCP group said they want to hear more about the ICA community and to contact the helpline to tell them more about the metadata and analysis need. And if I forgot anything, I invite everybody that participated to the breakout session to raise your hand. Thank you.
Thank you very much for that, Chloe. Yes, questions, Dana?
So related to the normalization, but something more basic that has been bugging me, that the immune population is one of the populations that have been typing with markers and sorting for a very long time, well before the HCA. And with CiteSeq, it's driving now a lot of the immune atlasing. And certainly for me that look a lot into cancers and some tissue residence stuff, what I found is a certain set of markers that you love and have analyzed to death in the immune organs. When I look at that combo of markers in even healthy tissue, not to mention cancer, the other genes are very, very different. And
that's true.
so what are you doing to avoid over-interpreting these classical immune markers that you know and love, and the normalization around CiteSeq to sort of really acknowledge the fact that maybe we have more unknown, and those markers, some of which are exactly that, markers, they're not even functional, might not be what we thought they are in some other contexts.
That's a great question. We didn't discuss this particularly, but I can provide an answer. And I should mention we did discuss a little bit the need of as part of the cell type definition, we need a validation and a functional annotation that goes with it, and it remains a problem of the scalability of doing high throughput functional assays. When we talk about the surface markers, we were trying to, we do need to engage a wider immunology community, right? So they're not ostracized by this whole effort, and they really adopt the data. Now, the idea is still to do an unbiased analysis and reclassification, and these CiteSeq markers can help guide in some extent.
They're good as long as they don't.
But they should not be driving it, right? It's like a way of knowing if these markers we've been using actually do track correctly with the new definition, and if they do, it helps you connect and connect with the existing data. I think we all agree on that. For sure. But we do believe that connecting it with existing flow data that has been published will help connect with the immunology community and kind of clarify what still holds and what doesn't hold.
Having the measurement is good as long as it's not over-interpreted.
Absolutely. Yeah.
One more question, maybe?
So, how much discussion has there been, which I've seen in the past, about the kind of malleability of immune cells? Because I know in previous sessions people were concerned about that, but now that people have measured a lot more data, how much do they look kind of stable to them?
In terms of when we sample from different tissues, autopsy, not autopsy, do they look? Somebody brought this up. We didn't do a deep dive in that discussion. I think there's a lot of data out there that shows that within conditions, you do see technical variation, and the hope is that by scaling up and seeing more and more cells, we'll be able to kind of figure out what was a technical artifact from what wasn't. But it's scalability that will tell us. Yeah.
Great. In the interest of time, I think we need to move on. So so next we have reporting on the lung, Fabian Theiss from the Helmholtz in Munich.
Thanks, Kirsten. So we had a discussion about the lung. Maybe you can bring up the slide. I was really happy that some people found their way up to this sort of exiting the whole place room, but I think it was very intense. It was definitely a nice organ to talk about. Thanks, here we go. So this was together with Martin and Herbert. We first sort of gave a short introduction, what we think could be points to discuss, and then I think we had quite lively discussion essentially about the following points, and I think I should use this thing here, right? Excellent. So we just reviewed briefly what you already saw in the nice introduction by Aviv and Sarah, essentially showing that there have been actually quite a few atlas studies around now where people have been aggregating cells at very different levels, up to now 200 individuals with more than 1 million cells profiled, very different protocols and so on. And we realized that across all of those studies, there's of course maybe even like some type of mosaic, as Martin so nicely formulated, of different studies. Data integration was still a bit of an issue. There is actually rather minimal spatial mapping that, although annotation for quite a few of those studies in the spatial setting has been provided. There's no consensus on actual cell type annotation, and there's very different ways of what people actually then sort of break down in terms of subcell types. So from the computation side, the whole mapping aspect of actually going on the hierarchical fashion, for example, or potentially also not finding overlaps, was a challenge we realized and discussed, and there's an unclear effect of how this not yet existing harmonization on experimental techniques is actually influencing that. We discussed a bit about that, which I will summarize in a minute. Notwithstanding that, actually now a few projects funded that actually put money now into generating new data sets in this line, and there's many people shared across those different projects, so they know about each other, and we've been discussing also how to bring parts of those data sets together. And I should highlight, we summarized all of those discussion points up there if someone wants to read up on that. So first point we discussed and spent quite some time on was how do we actually build now a coherent atlas? So how do we go beyond this mosaic of the different aspects? And first point, as comes up so often, I think, for the past 3 years now, where do you sample from, right? Do you sample localized, well-defined region? How do you actually communicate this well-defined, localized region? Can we just go into CCF coordinates? But obviously we realized it's a bit of a chicken and hen and egg, I think it's called. You know, right? Chicken and hen and egg, you know, this type of problem. Recursive issue. So you get the idea, right? You cannot... So how do you actually define those regions? So our conclusion, I think, from the audience, and please everyone in discussion help me if I'm commenting wrong on that, particularly Herbert was volunteering to jump in here, was that we first want to have good annotation of the various regions. There was even suggestion potential video tracking or using surgical devices when people actually remove tissue to potentially actually get more insights into that. And in addition, we discussed that obviously we're very interested in adding now more clinical covariates. Let them be within DCP or outside, but getting access potentially even whole genome sequencing or at least SNP arrays for each of those subjects. If those sizes increase as they currently do, this will become interesting very quickly. Remember the nice tetrahedron or covariate space that was shown, I think, down in the end. In your dive, you even actually superimposed a genetic variation on top of that. So it would be, of course, interesting to keep that. And in fact, some of the protocols are already regularly gathering this. In terms of protocols, we discussed whether it's meaningful to define an optimal protocol. Also, this question we had a few times already in former HCA meetings, and it was quickly agreed upon that this is not feasible. So you cannot just say, okay, this is the thing we do, and now everyone just goes according to that procedure. But instead, we might want to learn how to potentially map data from one procedure on the other or merge them. There was a suggestion from a more computer-affine person to do something called style transfer, which people have been doing in the vision community to potentially map things across each other, but that was a bit on the debated side, so I won't comment more on that, although I do think it's interesting. Sorry for that, Dana. But I think knowing how to map between those protocols is very interesting. And we should be, of course, very much aware that the variation in the preparation steps might completely overshadow those variations that you then do get in the end, whether you use 10x or SQL, right? Of course, this might be difficult to capture. And so we agreed on that it's very important to put those steps that you actually do in your protocol properly document and put them, let's say, in protocols AI. I think most of those studies are now actually agreeing to do so, and I think it's becoming more of a standard anyway. And this way you can actually annotate this meta information, potentially superimpose it on the overall integrated thing in the end, then learn what maybe some sources of variation are actually influencing this. So once we have the coherent lung cell atlas, the next point to discuss is, of course, how do you reach then a consensus on cell annotation and then ontogeny. This maps nicely to what Peter was presenting with, I think, the common annotation framework, as was nicely called. So also there we agreed there was suggestion that there's maybe the expert that annotates the data, and this would be the set of notation to go forth. In particular, the original one would be very important to have. But I think we also agreed upon the fact that it's important to then allow for variation, particular notation to one certain thing that you might be interested in this setting. And there was a sort of a strong feeling so the annotation has to be heterogeneous. There was a second step, actually, in this particular, it's one of the larger scale papers coming out by arXiv recently, was that the clustering in this particular one is done on the individual level, and then only on the marker genes and then start sharing annotations. That, of course, does away with this somewhat complicated data integration problem, but then on the other hand, it's also a bit of potentially too strong simplification. We were discussing which of those two ways to go. In the long run, we do see that an overlay notation of a jointly embedded thing would be the way to go. With respect to variation across humans, I think we all agree it's important to discuss and include this. I mentioned this before, according to HCA goals. At least I got together gender, age, ethnicity. I don't know if I remember, if I forgot something else. Those would be important to document. Geographic diversity. True. I just remember the map. But then some more subtle variation that might be particular to one state, of course, would be nice to document, but not so straightforward. And how to delineate that was something we discussed in the computation slides a minute later. And one question that came up again, we had this a few times before, is there older studies, potentially maybe on the bulk RNA side, but maybe also more so on the imaging side that were actually given whole organ imaging, particularly in lung, where the structure is rather regularized, to actually leverage those and then combine with newer data. And there are some experts, I think Kaminsky Lab was suggested as one of the contact persons to maybe interact with the CCF. Then a long discussion on programs and states. This I found actually very fascinating. There was discussion that we not only annotate groups of cells or regions in potentially also spatially resolved regions, but also groups of co-varying genes, potentially co-regulated genes, right? And that this old picture of yonder, namely gene ontology categories and programs, is a bit to be challenged, and that we could actually derive data-driven patterns, and doing this in a sort of more standardized, potentially way to actually lead into an updated ontology would be very interesting. And there's of course ideas in computation community around for quite some time, going into factors or maybe also nonlinear factor analysis. Let's say if you have a layered version such as neural networks, to actually make use of those. The last point was the common annotation framework. Of course, do we only focus on a single one? And how do we make sure that if someone is actually challenging one particular annotation, why he or she does that, and maybe then actually annotate, because I was not happy with this particular reason, and make use of this comment. That was a point was taken. And I think the last one that I didn't put on the slide, because we didn't actually write it down, was that if there's multiple of those existing data sets around, just giving access to those, but then also visualizing on the existing different sub-data sets what the changes are on a visual interface, such as the UCIC browser or others, was something people were asking for. There was discussion on the spatial data into Atlas. As of now, there's not much yet generated on an available fashion, but in all of those, so in particular those two H2A20 as well as the CCI seed networks, this is a big part. And one initial question was, in particular if you go on more targeted approaches, what is a good way to select a subset of genes to then go for, let's say, in your Murphy experiment? And purely computational design from experiments doesn't seem to be the only way to go. So that's an interesting experimental design question. With respect to resolution, some 3D approach, so 3D could mean very different things, as I was told, from just the real fine-grained few micron resolution to the more millimeter C-stacks, and how to bring those together. It's not clear. Experimental design, I mentioned already. I think we should learn particularly from space TX version, and on the whole spatial whole organ imaging, it's not clear how we bring this together with the CCF. So we decided, in particular with the short running times of some of those projects, to just go ahead. Last part, I think, was on the analysis. What do we do downstream? Should we process all data sets in the same fashion as we want to do in the DCP? Yes, we should. Accessibility, how do you actually put all of this, make it accessible? Well, this would be what the original projects only do, pending on DCP's agreement with GDPR and all those political legal issues, but we heard that this will be coming hopefully soon, and we're looking forward to that. Should we share embeddings or UMIS from the original studies? We felt not so much, and I think I mentioned already the regulatory part. How do we deal with the high throughput of imaging data? This is to be pending, and we will just focus it once we generate it. And then I'm done. How do we actually make use now of the atlas? This was, I think, a very interesting point. What would be the actual atlas use? And of course, this you would see, I think, in all of the 6 projects that you hear. But I think it was still nice for us on the computation side to learn what is done there. First, of course, you just want to map out the variation that you do see across the 3 or 4 different axes that should be age, gender, ethnicity, and spatial location. Or how do you call it? Regional. So in any case, we want to basically map out this variation, and if you now do your own study, you want to basically get tests. Are we within normal or not? Can we map this on top of it? So this baseline for each own DC studies, which are coming up now a lot, will be important, both on just simple differentiation, but also, let's say, if you have spatial data on cell-cell communication. And then the last part would be potentially interesting to get, once you actually have now made your atlas, to go beyond just doing your own t-SNE or UMAP again and again, to essentially have, for visualization or also then for downstream analysis, this whole latent space learning thing, which is a big thing in the computation community at the moment, to actually take latent spaces that have been generated across an agreement of many different data sets and then go with those latent factors to start with. This you could use for differential expression analysis and experimental design. That was it. Thanks.
Great, thank you. Right. Are there questions for Fabian?
I think it was in the previous slide that you said you weren't certain what to do with the CCF, so you were just going to...
Yeah.
Was it not do anything or collect something? It just wasn't clear.
So I think when we said where we have the spatial mapping, how we intersect this with the ongoing, sort of not yet finalized CCF endeavor, and there we said due to the short running times, we just go ahead and basically collect spatial localization, but not yet know how to directly map. I think it's just that's the most pragmatic way to go, and that's what people actually figure out.
Great. We're already slightly over time, so I think we'll move on to the next presentation, which will be from Alain Chediotal from the Sorbonne in Paris.
Hello, everyone. So I'm going to just present you what we discussed during the development session. And as you will see, it's still under development for sure. And I agree 5 minutes to prepare is a bit tight. Anyway, I'll do my best. So I also did the same kind of exercise like 2 years ago in Ixton. And something has changed, and some other things haven't changed in 2 years. So what has changed now is there are some initiatives around that are starting to be funded just for development. So I listed a few. So of course, we see the HDBR that already existed 2 years ago, obviously, but that got some more funding recently, so that's going to continue. There is this new initiative, HDBI, that is also related to data biology in human, that was recently announced and financed by the Wellcome Trust. There's still this Swedish Human Cell Atlas project that is also funded. And then we have in France now this UDECA project is also related to human development. There are also other initiatives in China and in Switzerland. So at least now there is some funding available and some project that will be running. So I think there will be a lot of progress in the next 2 years. However, we still think that the community doing development is not involved enough. So, I mean, if you ask here, we asked during the session, there are not many people who do development who participate to this HCA meeting. And that's really annoying, that's really frustrating. So I think there's a big effort to do to try to motivate them to attend the meeting, or that people from the HCA community attend the development biology meeting and tell them that it's important. So I really don't get it. I don't know why they don't think it's interesting or why they don't want to contribute. So it's really something that I'm really surprised not to see here more people doing development. So I think there's an effort on our side to try to to try to attract them here and to try to really tell them that it's something that is important. Obviously, funding can help, but that's not the only solution. So I think it's really something that is needed. So we were wondering, I was asking, so we asked people that are running the portal, where is development on the portal? And for now, it's not a dedicated entry. So it's like if you work on the GUT or something, then there will be development hidden somewhere. But maybe we thought it might be better if there's a specific portal or entry somewhere that makes clear that this is where the development data are. That's what they did for the humans, for the Allen Brain Atlas. There's there as well the development that has its own entry, and I think that's something that might give more visibility to this problem. So either it could be through the portal, or maybe we have to do something else on the side that is connected. But I think for the development community, that would be better if they could share like questions, advice, and what we need is to build a network. We don't even know who's doing like a developmental cell atlas in the world. There was a paper recently on the retina from Seth Blackshaw. No one in the field knew that he was working on it. So it's too bad that he's not here, that he's not involved. So I think there's really need to structure things, and then that where we might need to get some help, because again, that's really something that is needed. So one thing that might help is that there will be a meeting in Toronto in July 2020, so next year. So I think there's really a need to advertise it very, very well to the developmental community to try to have them attending this meeting. That will be the first one that is really dedicated to it. Hopefully there will be a next one in 2022 in Paris as well, but it's really something that will be important. So it's a good chance to promote this type of research. So I mean, we can learn from what they have done for the brand development part, which is much better structured, because also they got funding from the Allen Institute and in particular. So I think there we can also try to get advice from them or how we can build something that is working well like their own project. So as I said, we need really to have communication. That's really a big thing that is missing. And this already mentioned is what I said. We need to have a way to share the data better and to between the people who do development. So some things that are like, oops, is it one way or the other?
You have this is for the piece.
Oh, sorry. Okay. So one thing we said is there are two ways also of doing the thing. So we discussed this. Should we say, okay, I work on the gut, so then when I do the adult gut, I will do at the same time the developing gut? That's one way to do it. We thought maybe it would be better to try also to ask people who do development, what do you think are key questions that are still not addressed in human? What are key time points during development that you want to study for each organ? So a simple example we gave for a project that we do for EU is, okay, if you work on sex determination, you want to study it before sex is determined and right after sex is determined. So you will study it between 7 and 9 weeks or 10 weeks. There's no need to study it at birth or to study it at 14 weeks. So I think it will be also important that people for each organ, they identify key developmental time point and questions, rather than adding like, oh, let's add a 12-week-old embryo in my sample, and then we get something about development. And maybe there's no need to do the whole series of development if the question you want to address is just between 2 or 3 weeks of this, at some point in the gestation. So I think it's important to think, we said about the questions before, what are the important questions, what are the key questions in development you want to address, what will be the best time point, and how can you get the samples. And that's where you might need help from people who do development, because they have ideas, they know from what they've done in those models, what is really key here. So that's something that is really important. So we discussed about there are people working on neuroquest cells, there are people working on skin, on joint, on brain. So and for each system, model system or organ, there's a different time that might be more relevant. So it's really something that is important. So we said, should we stick to normal development? So we have the same kind of idea, also include disease. So for now, we kind of agree that people say if we don't have disease, it's harder to get funding, so which is true. There are some countries where if it's not disease-related, it's going to be very difficult. So after discussion, maybe one idea that someone suggested is maybe we don't include disease, but we could also try to focus sometimes on genes that we know are mutated in some developmental diseases and try then, if they're expressed in specific cell types, or if we know they start to be expressed at a certain time point, maybe prioritize by focusing on those genes that we know are linked to a disease. And if we understand better what the function is during development, maybe then we'll be able to know better what goes wrong into those diseases. So it's not disease exactly, but it's one way to still link it somehow to disease. But again, there's no consensus around this. But we think that for now, in terms of funding and effort, we cannot do all the diseases. There's so many developmental diseases that will be impossible. So we discussed, as before, about the variation between individuals. The one thing that we think is really important for development is really the female-male differences. So it's something that we discuss about people who do batch. For now, it's clear, even for the brain, male, female, everything is mixed. Okay, and we think it's not a good idea, at least for us. So I think it's really something that is important, at least for development, to really pay attention to sex-dependent variation. So if you work on sex determination, it's really obvious, but you might find interesting things if you pay attention to this in other systems. One thing also that is important is that the embryo or the fetus is not alone, if you wish. The mother is also there, and so there are also importance with the body mass index could have influence between obese mother and lean mother on the fetus. If the mother takes some drugs, medicines, if she drinks a lot of alcohol, everything has to be considered. And for now, when we get the samples, often we don't know the condition of the mother. So it's something that will be complex to add to the pipeline in terms of ethics, but it's something to think about, and that's something we're starting on our side. Can we get some information or include information about the mother herself? Because that's something that could have a strong influence on the way you think of a sample of fetus, an embryo tissue is normal or not normal. So for it, how can we tell? If it's an adult, you can tell most of the time by if the person was normal or not, but for an embryo or fetus, it might be difficult. And we don't have enough funding to do whole genome sequencing, and we couldn't do legally whole genome sequencing of all the fetal and embryonic tissue. So how do you define a normality in our cases? So that's something that is also important. So in terms of technology, one thing that is unique to human embryo or development is that we can have one individual in one data set completely. So we can have all the organs, all the cells, either by images or by sequencing, and that's not something you'll be able to do in an adult. So it's really something that we heard the gut is huge. If you take a 9-week-old embryo, it's not so big. So you can really sequence the whole gut, you can map cells across the whole gut. It's not a problem. So there's a lot of development that are interesting technicals that will be good. So you can really go to a higher level in embryos. So just a few things. So this is already mentioned. That's something I won't discuss more. And also there are some specific ethical concerns when you work with human embryos that are different from the adult. One, we work on dead individuals for sure. But then there are also some problem, depending on countries, legislations are very different. So it's also something we have to sort out with people who do ethics, and that's something we're doing already now at the European level. But what are questions that are specific to development? Okay. And so I will stop here. There are more techniques, and that's one example of the stuff we do. As I said, here you can have the whole embryo in one image, data sets. So it's really something you can do and that you cannot do in an adult. So it's also something that is interesting. Okay, I'll stop here. Thank you.
Great, thank you very much. Do we have any questions right now?
Just asking about your difficulties to get developmental people involved, and it's like a comment or a thought. What I feel, for example, for the heart, it would be really useful to talk all the time. Have a continuous exchange from the adult heart to the people that are doing developmental heart. So on one way, the way of integrating, I think it's important that you go also across organ per organ or system per system, not just keeping the developmental atlas as its own group. Do you know what I'm trying to say?
I agree. No, I agree. No, but I think there's really, to me, there's a problem. I don't understand. So we need to, there are some very good groups everywhere. Some of them are doing human, they're not contributing anything. So it's something, I mean, at some point, okay, someone has to do it. I will have to try to find out and try to contact people and try. I said maybe we need to have like a place where people can meet online, okay? So like a page, something that is better than what we have now. Last time, 2 years ago, we had a Google Drive that I wrote a text. I told people contribute, add something, add nothing added, 0. So I think this time we should try to find something that it works a bit better. So through networking or participating to the Ontology meeting and promoting this initiative. So.
Right, so one last short question because we are over time.
Oh, sorry.
Hi. So beautiful talk, and I just want to say a couple of things for the disease. I mean, the normal state can just be done by transcriptional karyotyping, right, from the single-cell RNA-seq for duplications and deletions. You can see that. So even without paying for WGS. More importantly, you know, these beautiful images. Really, what's your view, or what was the discussion on how it can be linked to the single-cell genomics and histological level? I mean, did you have a discussion around that? Is there a plan?
Just briefly at the end, it's clearly that's something we discussed with the need to link everything. So, but then there are some technical development that are needed. So it's part of what's should be funded by EU. Where normally that's the idea. Like, can we go from a 3D dataset like this, sample? And then process it further with like RNA-scope or other things. So can we combine things where we together?
Or cross samples. I think that would be awesome. It would be amazing.
It should be possible. But I think it's a time now that there's again funding. It's still like important. It's a time where we can start testing things and bringing people together to help us. Okay.
Great. I think we need to move on now. So our next presentation will be from Mena Clackworthy. From the University of Cambridge, and she's reporting back on the kidney breakout group.
Okay, so if we can get the slides up. So I'm reporting back on behalf of myself and Anna, and also the other participants. So we had some lively discussion around efforts relating to the kidney cell atlas. So what we did first of all was to just briefly review the data that's at present being submitted to the DCP. I think Sarah and Aviv presented at the start a summary of data that is badged as HCA data, which I think said that from a kidney point of view, there were samples from more than 60 individuals and 1 million cells. But actually, if you look at what's actually been submitted to the DCP, it's much less than that. So I guess that's the first point, a sort of call for us to get on and interact with the DCP to get our data up there. What was also obvious reviewing that data, which in part relates to some of the metadata that are associated with our samples, is that for much of this data, the precise location from which the sample was taken was not clearly identified, and so that's something for us to take going forward. We then went on to talk about some technical or legal challenges, and the first thing that we thought about, that if we're trying to generate reference data sets, and for example, one of the seed networks relates to this, so where we take samples from one center and share it across centers to allow different technologies to be tested. What are the challenges that we face practically to undertake that effort? And you know, the really mundane thing that's currently holding up those sorts of studies is of course MTAs between institutions, because this will involve multiple MTAs between different institutions. So we discussed there might be a number of ways of overcoming that, and one of those would be to actually upfront now perhaps try and set up MTAs that were for the Atlas project, so that there was a single MTA. Or at least that what we might be able to do to get to this point was to use templates that are already out there, so from existing networks. So for example, the KPMP, so an NIH kidney precision medicine project. So there they already have MTAs that are set up, that maybe we could use those templates to drive this forward as quickly as possible. And then another thing that we gave quite a bit of time to discussing was we've talked about sampling different individuals in terms of male, female, and also across developmental time and age, but also across different genetic backgrounds or ethnic backgrounds. And actually, practically, that really is a problem. So certainly in my own centre, we're limited in terms of the samples that we're able to access that are from non-European genetic backgrounds. But I think it's something that we need to address seriously. And from the point of view of the Kidney Atlas and going forward thinking about kidney disease, this is not just a matter of being politically correct. It is something that's significant and that as a clinician I see. So we know that individuals from different genetic backgrounds are much more susceptible to some kidney diseases and also much more susceptible to more aggressive and rapidly progressive kidney disease. And we discussed that in order to move forward in this regard, what we need to do is to have multiple sampling centres, and that if technologies become available where we're more easily and able to utilise frozen samples or even formalin-fixed samples, then that will facilitate us being able to take samples from across different groups. Then the next thing we went to was the practicalities of metadata, and the DCP have provided an outline of metadata that they require. But we also talked again about the fact that there are a number of already established kidney collaborative efforts that have spent a lot of time at developing frameworks for collecting metadata. And that we really need to utilize that, so not to reinvent the wheel, but to be using the same standards as are being used there. And ultimately, that this will better allow us to share data and to analyze each other's data. We talked a little bit about properly defining the anatomical position from which we get our samples. And again, at HubMap have some tools for doing this, and we heard that they have a 3D visualization tool, so a 3D rendering of a kidney where you can actually try and mark on that the exact position from which you take the biopsy. What we also said was that, and this could well be useful, and hopefully the kidney group are going to get a demonstration of this, but that human individuals, so if you've ever actually personally taken some kidneys to try and sample them, are very variable. And so even with a sort of standard 3D rendering, there's much more to be gained in terms of anatomical position than that. So we thought it would be a good advice in terms of standard practice to have a photograph of exactly where the biopsy is taken from. So if somebody just happens to have a lot of fibrosis in one area or a smaller upper pole, that we can see exactly where that sample has come from. And we also heard from Matthias the importance of us taking, wherever possible, paired samples for histological analysis so that we can relate it back to, the last 200 years or 100 or so years of literature defining kidney normality and disease. Then we talked a little bit about spatial technologies, and actually the kidney is really tricky to image, so the tubules are phenomenally autofluorescent, and that is a challenge for a number of technologies. So I guess we thought that at the moment this was an area that really needed more work, so in some ways was lagging quite far behind our efforts to generate certainly single-cell transcriptomes. And similarly, although there are a couple of efforts to try and use single-cell ATAC-seq on kidney samples, again, these are relatively in their infancy. Now, as previous groups, we also talked about the challenges of sampling rare cell types and strategies practically that we might use to do that. So one strategy is to flow sort larger groups of cells in which we think the rarer subset is contained. But that can be difficult because the process of flow sorting in itself can introduce bias, and there may be some cell subsets that don't deal well with the whole sorting process. And so, again, having parallel spatial data we thought would be helpful for that. We also discussed that magnetic bead selection is another way to get around that. And in the kidney, one thing that we've noticed is that if you generate a cell suspension, lots of the cells that you get in there are proximal tubular cells. So one way to enrich for rarer cell subsets is just have a selection step where you get rid of all the proximal tubular cells, and then whatever's left over will naturally have more of these rarer cell types. We just touched briefly on the fact that although cell hashing technologies are being trumpeted as being pretty useful for multiplexing samples, the data that's out there at the moment is from PBMC or blood. And actually, when we've tried this on kidney samples or tissue samples, it's much more challenging, and actually not all cells seem to get labeled or are double labeled. And we heard that there may be some newer technologies. So Wolfgang was telling us of barcodes that actually get inserted into lipid membranes. So don't rely on the surface expression of a particular protein or antigen. And we summed up by saying that as a network, we felt we could improve our communication and collaboration, and encouraged us all to sign up to the Slack channel. We also thought that these meetings are really useful because we actually go through an agenda, and we all have great experiences to share, and doing that more frequently might be helpful. And our CZI representative suggested that maybe a quarterly conference call would be a good idea, and that in these calls we could focus on particular areas, say how we've all got on with spatial technologies or with digestion protocols for each one. And then we also had highlighted that in March 2020 at NIH there will be a meeting for a variety of cell mapping efforts, and that perhaps we could use that as an opportunity to have a breakout group for the kidney mapping efforts and to try and discuss these things in person. And finally, I guess, which would integrate well with that meeting, is that we really should try and reach out to other communities that are mapping or generating cell maps of the kidney, both in health and disease, so particularly HubMap and KPMP, also rebuilding a kidney, so that together we can try and use standard metadata sets, metadata framework, anatomical framework, so that going forward we can really share our data and hopefully move ourselves forward in terms of using version 1 of the Atlas to think about, for example, disease pathogenesis, response to drugs, the questions that we've heard about being important.
Great, thank you very much, Mona. Are there any questions or comments from the audience? Just a short comment from me. We certainly also found that in digested tissue, cell hashing is a lot more tricky than maybe in PBMCs, so it's not that easy to implement. Any comments from the kidney community? No, I think in that case we might just move on. And our last but not least speaker will be Michaela Noceda from London. And she's going to report back on the heart session.
Hello. As I was telling my peers in the breakout session, the best organ ever. Although I have to say that sometimes I envy the people working with PBMCs who can access their samples very easily, while most of our samples, especially the healthy hearts, comes from people that have died or are about to die. So what I just mentioned before when Alain was talking, I think it's very important for us to coordinate with the developmental heart atlas. So the ultimate goal of most of us is really to study heart disease. But to do this, we have to definitely set up and create a reference atlas of the healthy heart, which is definitely not an easy task, but extremely exciting. So I just want to share with you where we are with the heart within the HCA, and thanks to Marion for sharing these slides. You see, these are projects uploaded from various organs, and in the heart we have two. Of course, the hemolymphoid are the most prolific of all, but hopefully we can increase very soon the contribution. The existing data on HCA are from human developmental heart and mouse. They were obtained from the States and the heart in the mouse, tabula muris, and in the human heart, we are talking about samples from Sweden. So there's not much there at the moment, but as I said, hopefully we'll be able to contribute very soon. Now, I want to stress something. I mean, the people that work on the heart know this, but I like to stress to the general, to all of you, what is the anatomy of the heart and why I want to share it, because the heart is one organ, but really is made of many different parts. Each part has a different anatomical shape, and but most importantly, a different function. So we have at least, we could say, 4 organs within an organ. We have the 4 chambers, we got a septum, we got valves, we've got little muscles that are able to open and close the valves, so the complexity is immense. And the organ is not a small the heart is not a small organ, talking about sizes, that seems to be. Each little area will have a different histology and likely a different function. So even if we focus on this area that we call the free wall of the left ventricle, it's a very thick piece of muscle. It's about 1 to 1.5 centimeters. So when you have to do single-cell of these or spatial transcriptomics, there are obviously some problems. And all of these cells are not identical. You have very important cells on the outside, the epicardium, and extremely important cells in the inside. And then in between, all an array of cardiac muscle cells, so which we know very little about the heterogeneity because they look very much the same if you look down the microscope. So what we came up with some important points and somewhat a plan. So the important is that we create a pilot set of data, a reference set of data, and most of us agree that these regions, the left ventricle, right ventricle, the atria, the interventricular septum, and the apex are the most obvious regions to sample. We all agree that the next phase should be hypersampling, or what we mean is trying to reduce the number, focus on a few donors, and really try to sample many different regions. At that point, we should have a good idea of where the variability stays, and then move on to a larger cohort and to assess even more the variability on selected regions of interest. Now, as I've been talking now, sampling is very fundamental. I mean, many of you today have brought up this issue. Location is not just a relative problem. It is a big problem. I mean, some colleagues from the broad said that even when they sample the heart with 1-2 centimeters apart, we see major variability. I mean, this is not a surprise. If you think to the atria is not very big, 3-4 centimeters, but the structure within an atria changes a lot, half a centimeter apart. So we can't forget these aspects that will give us variability. So someone has suggested, can we have a framework and define precise regions to be sampled? In theory, that would be really good, but in practice, it's hard. We use some bank samples, and for that, it's really hard to trace back where they were exactly sampled. For the existing ones, sometimes we can do it. In other times, if you are in a theater, time is ticking, you can't be too precise. But the general message is, please try to record as much as you can, and let's try to share this data eventually on the HCA. So I think the problem of tracing position today is coming across very importantly, and we should try to do something about this. From a picture for the gross anatomy to the histology, these points are very important. The size of the sample is important. As I mentioned before, if you think the left ventricle is really thick, so you want to incorporate all the cells from epicardium to endocardium, I tell you that's a big chunk of tissue that you have to process if you want to do that, or you have to section in smaller areas, and that amplifies the number of samples you are analyzing, or it will amplify your variability, because when you sequence 5,000 cells from a big chunk of tissue, then you're really somewhat randomly picking some cells. Then there was one comment that I liked. Are we sure we're capturing the expected variability? So there are some cases in which people have reported that with the current technologies, they're not able to capture genes expressed at very low levels. And in some cases, a gradient of expression throughout the chambers of the heart hasn't been captured. So in my opinion, we have to keep in mind that we need to run different technologies, not just the usual ones. In some cases, we'll have to sequence deeper and generate libraries in different ways. Then there is this picture that I use all the time, but I want to, people, especially computational biologists, and maybe they don't think about sizes of cells all the time. So we struggle with one issue. The cardiomyocytes can be really big, and they can be they are actually very big, and they can be binucleated. They can be about 100 microns in length. That's about 10 times the length of a lymphocyte. It might fit 20, 30 lymphocytes. So when we're doing single-cell analysis, we've got challenges. But also, I think we need to think when we do spatial transcriptomics of the heart. If the spots are 100 micrometers, we're going to chop up these cardiomyocytes in various parts. So if technologies are in the room and they're thinking how to develop new technologies, please keep in mind as it's not all about lymphocytes, but the cardiomyocytes have some challenges. At the moment, most people are relying on single nuclei to study the cardiomyocytes, but new technologies have been developed, and a number of technologies are being tested at the moment to try and get information from the entire cell. Now, one last slide, because I know we want to listen to the keynote talk, is something that, again, came up a lot today. But I like to make sure we can do something in regard. So integration of data. Of course, we said over and over on different HCA meetings that we need to share technical metadata. Of course, clinical metadata are important, I think are fundamental as well, but this is almost an infinite space in some cases. The information that we can have and in theory are available for patients is huge. Think about us that we use donor hearts. There are big files of data about these patients. So can the HCA, or how can we streamline the transfer of this data to the DCP? But I'm also talking about the clinician that has the data, then there is the biologist, and then there is the HCA. So we need to be able to streamline, help the clinician to transfer the data to us. I think Korush was talking today, they get PDFs, pages and pages of PDF. How do we transfer that in a table that can be used by computational biologists? I think it's a very important aspect. Another one that I really would like us to think about, how to integrate histology, histopathology, photographic records, others have mentioned that today, are important information that we need to be able to transfer and collect for the HCA, because they're going to be essential to interpret data. Thank you.
Great, thank you very much, Michaela. Any more questions or comments? Fabian.
Thanks, Kirsten. I was wondering, on the heart, there's a lot of also computational 3D models that people have been setting up and also using sort of to study across variation across populations. Does it make it easier to develop a common coordinate framework in this setting?
Definitely. I think that would be really, really helpful.
So is that already done? What's the state of the CCF and heart?
Sorry. Yes, there's a number of us working together and
With
creating.
3D modeling.
Yeah.
I just want to touch on your last point. I think part of it is, it's a PDF and so on, but I think the main problem is the abstraction problem. It's the level at which the clinical record is written is not really fully consistent for many medical systems.
Yeah, so basically, how do we transfer? The numerical system can be different, the units, etc., but that should be easily transferred.
It's the terms and they're
exactly.
Not, and it's I think very complicated.
Languages
and big problem, and it's something to pay attention to in our community.
But we should think about it and try to find a solution, step by step. Maybe creating, a minimum data set, and then build over that. Thank you.
Great. Any more questions? So I think that brings us to the end of this session then, and I think